Usually medicine feels, a bit like an X-ray. You see the broken bone, you set it, you fix it. It's very structural. It's visible.
Right, exactly.
But a massive new 2026 paper in the journal Cell just proved that when it comes to how viruses and bacteria actually manipulate our bodies, we are essentially fighting this completely invisible war.
Yeah, we really are.
So today, we are doing a deep dive into a concept called the eORFeome, and we're going to take a close look at a newly cataloged arsenal of microscopic weapons.
It's fascinating because the paper, it really does read like a captured alien espionage manual. For decades, the scientific community has known that pathogens, so, whether that's a virus, a bacterium, or a parasite, they hijack our biology by injecting these highly specialized proteins directly into our cells.
And we call those proteins effectors, right?
Exactly, effectors. And thanks to modern genomic sequencing, we have millions of these effector genes mapped out. Technically, in genetics, they're called open reading frames, or ORFs.
Okay, open reading frames. So we have the blueprints.
We have the blueprints, yes. But having the genetic code and actually understanding what it does, those are two very entirely different things.
Right, because we don't actually know what most of them do we? Like, we just know they exist.
It's actually a staggering blind spot in biology. For about 93% of these known pathogen effectors, we have absolutely zero functional annotation.
93%.
Yeah, 93%. The genetic sequence is just sitting there in a database, but we have no clue what that protein actually physically does once it gets inside a human cell.
So if you're listening to this, you have to picture a massive armory of unknown biological weapons. And that is basically the core mission of our deep dive today.
Yep, that's the setup.
The researchers behind this Cell paper essentially built a catalog for this armory. They gathered 3,835 of these completely mysterious viral, bacterial, and parasitic effectors into one giant library, which they call the eORFeome.
The effector ORFeome, exactly.
But, I'm struggling to picture the logistics here. If you have nearly 4,000 completely unknown alien proteins, you can't just put them in a petri dish one by one and stare at them under a microscope. I feel like that would take a literal lifetime.
It would. It would take decades. So they had to use a high-throughput method. It's called a pooled lentiviral barcoded screen.
Okay, pooled lentiviral barcoded screen. That's a mouthful. Break that down for us.
It's actually a beautifully elegant piece of bioengineering. So they took all 3,835 of these unknown effectors and packaged each single one into a lentivirus.
The lentivirus.
Right. You can think of a lentivirus as just a microscopic delivery vehicle. Its entire purpose is to drop a genetic payload into a cell.
Okay, got it. Delivery trucks.
But to track what's happening, they tagged every single effector with a unique sequence of DNA.
A barcode.
Oh, I see. A barcode. So you know exactly which delivery vehicle is carrying which mysterious effector.
Precisely. But wait. If you dump 4,000 different viral proteins onto a dish of human lung cells, wouldn't a single human cell just get invaded by like 50 different viruses at once? It sounds like you'd just get absolute noisy chaos.
You would. You absolutely would if you didn't perfectly control the volume. And that's why they infected the human A549 lung cells at a very specific, incredibly low multiplicity of infection, or MOI.
Multiplicity of infection. Okay, how does that work?
It's just a ratio. By keeping the ratio of viruses to human cells incredibly low, say 1 virus for every 10 cells, they rely on Poisson distribution mathematics to ensure that the vast majority of cells that do get infected receive exactly 1 effector.
Ah, okay. So 1 delivery vehicle parks at 1 cell.
Exactly.
So you end up with a massive pool of human cells, but each one is harboring just 1 single alien gadget.
And crucially, those gadgets start off dormant.
Oh, they do?
Yeah. The researchers engineered this entire system so that the effectors are only turned on when a specific drug, doxycycline, is added to the culture.
Okay, so it's a switch.
Right. Before the doxycycline goes in, the cells are perfectly fine. Once it's added, the effectors wake up and the manipulation begins.
Okay, so you flip the doxycycline switch and microscopic chaos ensues across millions of cells.
Yeah.
But how do you actually measure that chaos? You have to somehow figure out which protein is messing with which part of the cell.
So they solved that by engineering the human cells to have fluorescent reporters. These reporters are linked to specific cellular alarm systems.
Like glowing reporters.
Literally glowing. If a certain cellular defense pathway gets activated, the cell literally glows green.
Wow, okay.
But if a pathogen's effector successfully shuts that immune pathway down, the glow completely fades out. The cell goes dark.
That is so clever.
It really is. So they run this massive pool of cells through a machine called FACS, that's fluorescence activated cell sorting.
FACS. And how does that machine work, like, mechanically?
It uses fluidics to pass the cells single file through a laser beam, millions of cells one by one. It analyzes them and physically sorts them based purely on how bright they are glowing.
That's brilliant. You sort the cells into buckets of who is glowing and who is dark, and then you just sequence the DNA barcodes inside the dark ones to see which specific effector turned the lights off.
The efficiency is just unmatched. It allows you to test thousands of interactions simultaneously. And when they ran this screen, they uncovered incredibly specific mechanistic details for how pathogens sabotage our biology.
Which gives us a great roadmap for the rest of this deep dive. So, for everyone listening, we're going to unpack the exact molecular mechanics of how these effectors sabotage three major cellular systems: the NFE inflammation alarm, the P53 tumor suppressor guardian, and the MHC-I immune display window.
Those are heavy hitters in human biology.
Let's start with subtopic 1.
Yeah.
Hacking the inflammation alarm, the NFP pathway.
Right.
This is basically the body's frontline detection system, isn't it? When something foreign enters the body, NFP is the siren that blares, telling the cell to pump out inflammatory cytokines.
Exactly. It calls in the immune cavalry. It's a critical pathway, which means pathogens have a huge vested interest in shutting it down.
Makes sense. So what did the screen find?
They specifically looked for effectors that turned off the green fluorescent glow after they stimulated the cells with an inflammatory trigger called TNF-alpha. And one effector showed up as a massive hit. It's a protein called RAVE, R-A-V-E.
RAVE. And the paper says RAVE comes from Legionella pneumophila, which is the bacterium responsible for Legionnaire's disease.
That's the one.
So to get down to the exhaustive details here, how does this one single protein silence the entire inflammation siren?
To understand the sabotage, we really have to look at how the alarm works normally. Under normal conditions, NFD is held inactive in the cytoplasm of the cell.
So it's basically asleep.
Yes.
Yeah.
And it's restrained by an inhibitor protein called IB.
Okay.
When a threat is detected by a receptor on the outside of the cell, like the TNF receptor, a signal is sent inside to destroy that IB inhibitor. Once that restraint is destroyed, NFB is free to rush into the nucleus and sound the alarm.
So the inhibitor is basically a set of molecular handcuffs. And to sound the alarm, the cell literally has to break its own handcuffs.
Yes, and RAVE's entire job is to prevent those handcuffs from breaking. When RAVE is present inside the cell, EB never degrades.
Oh, wow. It just stops the process cold.
It does. But the researchers, they wanted to know the exact molecular target. How is RAVE doing this?
Right.
So they used whole cell mass spectrometry. This is a technique that essentially breaks all the proteins in a cell down into fragments, flies them through a magnetic field to weigh them, and tells you the exact abundance of every protein.
To see what RAVE was actually binding to.
Yes, and it turns out RAVE specifically targets and downregulates a human host protein called RIPK1.
RIPK1. Okay, let me try to visualize this for the listeners. Let's use an analogy.
Go for it.
If the NFP system is a blaring siren on the wall, and the motion sensor outside the building is the TNF receptor, RIPK1 would be like the specific electrical wire connecting the motion sensor to the siren.
That is a highly functional way to picture it. RIPK1 is an essential kinase sitting right downstream of the receptor. It's the wire.
So RAVE isn't a bomb that blows up the whole room. And it doesn't even touch the siren itself. It's just targeting that one specific RIPK1 wire.
Exactly.
But how is it physically cutting the wire? Do we know if the protein has some kind of structural mechanism that explains it?
Initially, no. RAVE had absolutely no known structural domains. It was a total mystery.
So how do you solve a structural mystery like that?
The team used an AI tool called FoldSeek.
Oh, an AI structural tool.
Like AlphaFold.
Very similar.
Oh.
They took RAVE's predicted 3D shape and ran it against a massive database of all known protein structures. Instead of just looking at the linear amino acid sequence, the AI compares the actual 3D folds.
And what did the AI find?
It revealed that RAVE structurally belongs to the TK superfamily.
The TK superfamily. Which means what, exactly, in a biochemical sense?
It means it functions as an esterase, or a protease. Essentially, it's an enzyme designed to cleave chemical bonds.
So it literally is a pair of molecular scissors.
It really is. The researchers even pinpointed the specific amino acids acting as the blades of the scissors. No way. They found the actual blades.
They did. They are glutamate 19 and histidine 21, commonly referred to as E19 and H21.
That's incredible. But how do you prove that those two specific amino acids are doing all the damage out of the entire massive protein structure.
You mutate them. The researchers went in and changed just those two amino acids, E19 and H21, into completely different molecules.
And left the rest of the RAVE protein intact.
Completely intact. And the result was immediate. RAVE completely lost its ability to inhibit the NF key pathway. The cell's alarm worked perfectly again.
That is wild. 2 single amino acids out of thousands are the literal difference between a cell successfully fighting off Legionnaire's disease and being completely compromised.
Biology is incredibly precise.
Let's do a quick mini recap just to lock this in. So bullet point 1, the eORFeome is a 4,000-strong library of these totally uncharacterized pathogen tools.
Check.
Bullet point 2, we can screen them simultaneously using barcoded lentiviruses and the FACS sorting machine. And bullet point 3, RAVE from Legionella snips the NF key alarm wire by targeting the RIPK1 protein using its E19 and H21 catalytic scissors.
Perfectly summarized.
So here is a question for you listening right now. If you were engineering a cell, say, to ignore inflammation for a therapy, would you rather destroy the entire siren or just chemically snip the wire like RAVE does? Think about how highly targeted therapeutics could use this exact trick to stop autoimmune diseases without massive side effects.
The precision is exactly what makes these effectors so incredibly valuable to study.
Absolutely. Okay, let's move to subtopic 2, sequestering the guardian. The p53 pathway.
Now, this is where the plot really thickens.
Yeah, because if you follow cancer biology at all, p53 is the absolute superstar. It is literally known as the tumor suppressor.
The guardian of the genome.
Right. Its job is to detect severe DNA damage or uncontrolled replication. If it finds a problem, it either pauses the cell cycle to fix it, or it triggers apoptosis, meaning it tells the cell to self-destruct for the greater good of the body.
And if you are a virus that relies on hijacking a host cell to replicate, the host cell triggering a self-destruct sequence is your absolute worst-case scenario.
Game over for the virus.
Exactly. Pathogens must neutralize p53. And we actually already have a very famous example of this in high-risk human papillomavirus, or HPV.
HPV, right, the main driver of cervical cancer.
Yes. HPV uses a protein called E6 to bind to p53 and flag it for complete degradation by the cell's garbage disposal system. It essentially assassinates the guard.
The E6 protein is a literal assassin. But when the team ran their eORFeome screen looking for new p53 inhibitors, they didn't just find another assassin, did they?
No, they found something from a totally different virus, human herpes virus 6A, or HHV6A, and it takes a completely different approach.
What did they find?
They identified a novel effector in HHV6A called the U14 protein.
U14. Okay.
To test it, they used a chemical drug called nutlin. Nutlin artificially stabilizes and stimulates the p53 pathway in the cells. It forces the alarm to go off.
And I'm guessing the U14 protein potently shut that pathway back down.
It did. But the big surprise came when they looked closer using immunofluorescence imaging.
Let me guess. They thought the p53 would be gone, destroyed like with HPV.
Exactly, but it wasn't. U14 was not degrading p53 at all. The p53 protein was still highly abundant inside the cell.
Wait. If the guardian is still alive and present in the cell, why isn't it triggering the self-destruct sequence?
Because U14 physically restrains it?
Physically restrains it.
Yes. The imaging clearly showed that U14 binds directly to the specific DNA binding domain of p53.
Oh, the part that actually interacts with the human genome.
Right. And by grabbing onto that specific domain, it physically drags p53 into these dense clumps inside the nucleus, which are known as nuclear foci.
Nuclear foci.
Yeah. It essentially barricades p53 in these clumps, completely blocking it from ever making physical contact with the host's DNA.
Wow. Okay, so if HPV-E6 is the assassin that totally eliminates the p53 guard, HHV6A's U14 is a kidnapper.
I love that analogy.
Right. It just grabs the guard, throws them in a closet in these nuclear foci clumps, and locks the door. The guard is technically alive, but completely blind and totally useless.
That captures the mechanism perfectly, but honestly, the biology of how it actually binds is truly mind-bending.
Oh, what do you mean?
When the researchers mapped exactly where the U14 protein interfaces with p53, they found it binds to the exact same molecular pocket as a completely different viral protein.
Really? Which one?
The large T antigen from the SV40 virus.
Hold on, wait. Are SV40 and human herpes virus 6A from the same viral family? Do they share some ancient ancestor?
Not at all. They're completely unrelated viruses with entirely different evolutionary histories.
Right.
SV40 is a polyoma virus. HHV6A is a herpes virus.
Okay, so how do they have the same mechanism?
Convergent evolution. Over millions of years of random mutation and natural selection, they both independently invented a molecular tool that fits like a key into the exact same lock on the human p53 protein.
That is an unbelievable example of convergent evolution. Two completely different blueprints building the exact same molecular handcuff for p53.
Stunning.
Okay, let's do our second mini recap. So bullet point 1. Pathogens must neutralize the p53 tumor suppressor to keep the cell alive so they can replicate.
Correct.
Bullet point 2. HPV's E6 protein accomplishes this by completely degrading p53.
Yep.
And bullet point 3. HHV6A's U14 protein uses convergent evolution to simply sequester p53 into useless clumps, leaving it intact but totally blind.
Nailed it.
And that leads perfectly into an audience question. Why might it be evolutionarily advantageous for a virus to merely sequester a host protein like U14 does, rather than expending the energy to completely destroy it like E6 does?
It's a great question about the economy of a cell. Sometimes a reversible kidnapping requires a lot less biological energy than a full-blown assassination.
It's a dark sort of efficiency.
Very dark.
Okay, so we've seen Rave snip the alarm wire and U14 kidnap the security guard. But the human body isn't just a single alarm system. What happens when a virus needs to evade multiple, completely different security networks at the exact same time?
That's where we get into subtopic 3, multi-tools and de novo inventions. The paper dives deep into how effectors handle the STING pathway and the MHC-I pathway.
Okay, remind us what those two pathways do.
Those two pathways are essentially the pillars of your cellular defense. The STING pathway is part of your innate immune system. Its job is to detect loose, double-stranded DNA floating in the cytoplasm.
Which usually shouldn't be there, right?
Exactly. Loose DNA almost always means a virus is actively replicating in the cell. So STING detects it and triggers a massive, localized inflammatory response.
Okay, so STING is the internal smoke detector. And what about MHC-I?
MHC-I is a critical part of your adaptive immune system.
Right. I always picture MHC-I as a display window on the surface of the cell.
That's a perfect visual.
It takes little fragments of proteins from inside the cell and literally holds them out the window for passing killer T cells to inspect. If a T cell sees a viral fragment in that window, it knows the cell is infected and destroys it.
So STING is the internal smoke detector, and MHC-I is the distress flag hung outside the window.
And a smart, persistent virus needs to disable both of them to survive long-term.
Exactly. And the cell paper highlights how human herpes virus 7 achieves this using a protein called U21.
Now, prior to this study, we already knew U21 was involved with MHC-I, didn't we?
We did. It was already known in the literature that U21 binds to MHC-I molecules and diverts them to the cellular trash, the lysosome, before they ever reach the cell surface.
So it essentially pulls the distress flags off the window.
Right. But the massive barcoded scream revealed something completely new.
What did they find?
It proved that U21 is doing double duty. It also potently degrades the STING pathway.
Wow, it's a dual threat. Here's a mnemonic for you listening.
Yeah.
U21 gives you a 2-for-1.
I like that.
It's one single viral protein, but it crushes two entirely separate immune pathways.
It's incredibly efficient.
But wait, does it use the exact same part of its structure to attack both pathways? Like a blunt instrument?
It doesn't, and that is what makes it a remarkable multi-tool. The researchers found that U21 actually uses distinct structural domains for each job.
Really?
Yeah. A specific molecular tail on the U21 protein is required to drag STING to the proteasome and lysosome for degradation.
The garbage disposals.
Exactly. While a completely different structural interface on the U21 protein handles the MHC-I diversion.
That is so highly evolved. A true multi-tool.
It is. But speaking of evolution, that brings us to the discovery in this paper that absolutely fascinated me the most.
Oh, I know what you're going to say. The 13.6K protein.
Yes. The 13.6K protein from human adenoviruses, the de novo invention.
Okay, tell us about 13.6K.
This is just a profound mechanism. The researchers discovered that 13.6K, which is also sometimes referred to as the I-leader protein, is a shockingly potent inhibitor of MHC-I.
So it stops the display window.
Yes, and it is actually better at hiding the cell from the immune system than the classic adenovirus immune-evading protein we've studied for decades, which is called E319K.
Wait, so it's hiding the cell better than the gold standard we already knew about?
Which better?
How did they figure out the exact mechanics of what 13.6K is doing?
To map the exhaustive details, they utilized something called a fluorescent peptide assay.
Okay, let's break that down. A fluorescent peptide assay. So they synthesized tiny, glowing protein fragments to track where they travel inside the cell.
You've got it. Normally, little protein fragments are pumped from the main body of the cell into the endoplasmic reticulum, or ER.
The ER is like the cell's manufacturing and packaging center.
Right, and the machine that pumps them in is called the TAP transporter. Once inside the ER, those fragments are loaded onto the MHC-I display flags before they are sent up to the cell surface.
Okay, so the TAP transporter is basically the conveyor belt moving the goods from the factory floor into the loading dock.
That's a perfect analogy.
So what does 13.6K do to the conveyor belt?
Using those glowing fragments, the researchers proved that 13.6K is an ER-associated membrane protein that physically jams the TAP transporter.
It jams the conveyor belt.
It completely jams it. And with the TAP conveyor belt jammed, no peptides can enter the ER.
And if no peptides enter the ER, the MHC-I display flags remain completely empty.
Yep.
And an empty flag can't be sent to the cell surface.
Exactly. The cell essentially becomes functionally invisible to passing killer T cells. It acts as a highly effective stealth cloak.
That is mind-blowing.
It is. But honestly, as impressive as the mechanism is, the origin story of the 13.6K protein is the real marvel here.
Yes, this is the part that sounds straight out of science fiction.
Yeah.
Because 13.6K didn't evolve slowly from some older viral protein, did it?
It didn't. 13.6K is only found in primate adenoviruses. It does not exist in older viral lineages.
Where did it come from?
It evolved completely de novo, meaning from scratch, due to a literal genetic accident.
An accident.
It emerged from an alternative splicing event in the viral RNA. The most stunning part is that the genetic code for 13.6K is actually situated on the exact opposite DNA strand of a highly essential viral polymerase gene.
Okay, I really want to make sure everyone grasps the scale of what you're saying. Imagine you have a critical, ancient recipe for baking a cake.
Okay.
That's the essential polymerase gene. The virus absolutely cannot afford to mutate that recipe, or it dies.
Right, it's essential.
But somehow, by sheer chance, the cellular machinery started reading the back of that recipe card in reverse and accidentally discovered the perfect instructions for building an advanced stealth bomber.
That is precisely what happened. It invented a brand new, highly potent immunoevasion protein just by reading the reverse strand of an existing essential gene.
That is absurd.
It beautifully illustrates how rapidly and creatively pathogens can evolve new functions, even when they're confined to incredibly limited genetic real estate.
Let's do our final mini recap before we wrap up. So bullet 1, U21 is a 2-for-1 multi-tool blocking both STING and MHC-I. Bullet 2, adenovirus 13.6K acts as a stealth cloak by jamming the TA transporter conveyor belt to stop antigen display.
Yep.
And bullet 3, 13.6K evolved completely from scratch on the reverse strand of an existing gene.
Exactly.
Which brings up a massive question for you to ponder. If a simple virus can accidentally invent a masterclass stealth cloak just by reading an old gene backward,
Yeah.
what does that say about the hidden, untapped potential sitting right now inside the dark matter of our own human genome?
It strongly suggests the biological universe is far more flexible and holds far more potential than our current models even account for. And if we connect this all back to the bigger picture, it fundamentally changes how we should view these pathogens. We shouldn't look at the eORFeome merely as a catalog of disease mechanisms. The authors of the paper explicitly refer to these effectors as, quote, evolved perturbagents.
Evolved perturbagents, meaning they aren't just blunt biological weapons. They are highly refined tools designed to perturb specific systems.
Exactly. For billions of years, pathogens have been running the ultimate relentless research and development program. Through constant trial and error, they have evolved specific molecular scalpels.
Scalpels that modulate human biology with a level of precision that makes our best synthetic pharmaceutical drugs look like crude hammers.
Yes. Our drugs are often messy. These viral effectors are near perfect.
And that brings us to the ultimate takeaway from this research. We started this deep dive talking about an alien espionage manual full of unknown gadgets.
We did.
But now we see the specific utility of those gadgets. We know about proteins that can perfectly shut off inflammation like RAVE.
Right.
We know about proteins that can cleanly lock away a cell death trigger without destroying it, like U14. And we've seen proteins that can render a cell completely invisible to the immune system, like 13.6K.
The paradigm absolutely shifts from simply asking, how do we block these weapons, to asking, how do we harness them for our own use?
Exactly. Instead of just fighting these effectors, what if we harvest them?
It's the logical next step.
What if the cure for a severe autoimmune condition or the blueprint for engineering completely invisible CAR-T cancer therapies, isn't something we have to invent from scratch in a pristine lab.
The code might already exist.
What if the perfect molecular tool is sitting right now, completely unannotated, inside the genome of a virus?
It's a very real possibility.
So take that thought with you. The next time you think about viruses or bacteria, try looking at them not just as invaders, but as microscopic pharmacists who have spent 1 billion years doing the hardest engineering work for us.